Okay, good morning, everyone. Today we are going to be just conducting a review of the first 6 weeks of the course ahead of Wednesday's midterm and going through some details about the midterm as well. This probably won't take the full 50 minutes. It might, depends on how many questions kind of pop up along the way or how slow I go. But yeah, feel free to interject if you have any questions about the material. I also have office hours after this if you guys want to go through anything in a bit more detail. If not, if I do finish early, then I'll hang around at the end in case. I appreciate not everyone can always make office hours, so I try to leave a little bit of a gap as well. Okay, so in the first 6 weeks, we've learned about 3 main different methods of modeling evolution. We started off with population genetics. We then went on to evolutionary game theory, and then recently we've looked at adaptive dynamics as well. And adaptive dynamics, we'll be carrying on looking at that and applying it to some different scenarios over the next few weeks. So I wanted to spend today just recapping the main ideas as well as going through some information about the midterm to make sure everyone's on the same page. So just a few details about the midterm itself. It's going to be held in this room, AQ 5018, and it's going to be on Wednesday. And it'll be 90 minutes long, 10:30 till 12. Okay. So there's going to be 3 questions worth a total of 50 marks. Actually, that's slightly incorrect. I changed it. I think it's worth 60 marks. Not that it really matters. There are 3 questions anyway. And this midterm will constitute 20% of your final grade for the course. And the exam is going to be the same for the 495 and 795 versions. Now, any of the material that we've covered so far, in principle, can appear on the midterm. And the kinds of things that you're going to be expected to do are being able to derive equations or results that we've shown in class. Also, some of the sort of things that we've discussed in class. So make sure you go back over those notes. You'll be expected to solve problems similar to the ones that we've done in class, as well as ones that are on homework problems. And you'll also be asked to interpret results or the assumptions that go into our models biologically. Okay, so make sure you're familiar with how to derive some of the results, solve problems, and interpret. Key thing, you don't need to memorize any equations. Okay, so I generally don't like including memorization on exams because that doesn't test anything other than when you can remember something. I don't think that's a particularly good skill to test. You don't need to memorize anything. There is one equation that is not on the midterm, like as in it's not on the formula sheet or is not given to you in the question, but it is a very simple one that you can derive from the things that we have in there. So you shouldn't need to worry about any memorization. This is a closed book exam, partially because I'm expecting you to be able to derive things that we've done in class, and I don't want to just be able to copy them out from your notes. You can bring a calculator with you. You should bring a calculator with you. It should be non-programmable, non-graphical. And just as a general policy, as with all courses, there'll be strong penalties for academic dishonesty, up to and including 0 for this, potentially worse, 0 for the whole course. Okay. If you have any extenuating circumstances, if you're ill, if you have an emergency, especially if you're ill, please do not come. Let me know. Please contact me as soon as possible by email. I can make other arrangements. There won't be a makeup midterm, but I can transfer the weight to another aspect of the course. Okay, so I can rebalance things if you're unable to make it. Any questions about anything sort of logistic-wise related to the midterm? Yes.
So like grad students will have like in the science we had undergrad students and like grad students get like separate questions almost. So
yeah,
similar to
it is the same set of questions here. I might ask the TA to mark the grad ones in a slightly harsher way. So my expectations for I might be slightly more lenient in terms of the marks for the undergrads as opposed to the grads, but it will be the same set of questions. There are other aspects of the course, like from the project, for example, that the grads will have something different. They might have something different on the final as well. Yeah. Any other questions? Okay. So let's start going through the material. So the first 3 weeks we focused on population genetics. Okay. So We went through a lot of terminology during this period. In fact, I've got terminology on the next slide, so I won't delve into that just a second. We started off by focusing on haploid models and then diploid models. We started off as well looking at just a single genetic locus with and without selection. And then we expanded this to think about multi-locus models and how we can model the genetics at multiple loci and think about processes like recombination and something called linkage disequilibrium. Again, I'll cover all these terms again in a second. We also looked at ideas about dominance, how that can affect selection, the rate of which, say, an advantageous allele spreads through a population. And we also explored the balance that can occur between mutation and selection. Okay. We also discussed a number of applications of, why do we do population genetics? This can tell us everything from understanding frequency of alleles in the absence of selection, so, like a null hypothesis of what we would expect to happen, to things like how can recombination break up favorable or disadvantageous combinations of alleles, and also the rate at which, say, dominance can affect the spread of an allele through a population. Okay. So what I'm going to do is I'm just going to recap a lot of the key terminology that we've gone through. If at any point, though, you want to ask any questions or clarifications, please do. Okay, so one of the key things that we met, first of all, was an allele. If you remember, an allele is essentially just a variant of a gene at a particular genetic locus. Okay, so we often talked about capital A versus little a. There's nothing other than convention why people tend to use a capital A and little a. It could be anything. Okay, so these alleles, they are at the most basic, they could be just a single mutation. You can think about your nucleotides within your DNA, and this could correspond to, say, a G, and this could be a C, just different nucleotides. They could be combinations of these that always occur together. But as far as we can say here, it's just a variant of a particular variant at a particular loci. Okay. And locus, which is the singular, and loci, which is the plural, well, that's just telling us about the alleles that can occur at that particular location. So that particular locus might correspond to a particular phenotypic effect, like your eye color, whereas a different genetic locus might correspond to your resistance to disease or something like that. Okay, so we often refer to, we'll get onto genotypes in a second, but say we've got a genotype AI, BJ, AK, EM, then say this here is one locus, this first locus here. Note, because this individual is diploid, this same genetic locus appears in two locations within the genotype, okay? So this slash here is indicating that this is, this individual is diploid, so they have two copies of each chromosome. So although this is a single genetic locus, this is the same locus here. Whereas if I was to refer to the A and B loci, then this would be plural loci, okay? Is that clear? Good. So I just mentioned genotype. This whole thing here would be considered a genotype. You can think of that as being all the genetic material. Of course, individuals have way more loci than this in reality, but we might only care about a few loci for a particular thing. So we might think about the alleles or the genetic loci and the alleles that correspond to those genetic loci that correspond to a particular trait, like sickle cell or like eye color. So we might genotype people according to those. You can think as well, things like your blood type. We don't characterize you by your whole genome when it comes to your blood type. There are just distinct blood types which correspond to particular genes. So that's your genotype. Your phenotype is here. So your phenotype you can think of as being things like eye color. I mentioned other characteristics like disease resistance as well. So you can think of your genotype as the underlying genetic details, and your phenotype is kind of what's presented at the organismal level, typically. Okay, so your phenotype could be your eye color, your hair color. It could be things like how fast a cheetah can run. It can be preferences in terms of mates. It could be the biased production of gametes. All of these things essentially is some manifestation of what is going on at the genetic level. Phenotypes also actually have an environmental component. So things like your height is not just determined by your genetics, it's determined by, the kind of environment that you've grown up in. You've been able to gather enough resources to grow properly. Okay, so there is an environmental component there that we've not really talked about much, but phenotype, think about as your traits and characteristics. Genotype is the underlying genetic details. And then haplotype. Here, haplotype is just, in the case of a diploid, is just one sort of collection of alleles. So they've got two copies at each chromosome. So the haplotype here is just going to be, they have two haplotypes. They've got an AKBM haplotype and they've got an AIJ haplotype. And sometimes when we're modeling diploids, it's more convenient to model haplotypes than it is to model individual alleles. Okay, so I've talked about diploids a little bit. We covered briefly this concept of ploidy in general, and ploidy is saying essentially the number of copies of a chromosome that an individual has. Usually we refer to the number of chromosomes as N. So haploids usually have N chromosomes just by convention, whereas diploids have 2N, triploids 3N, tetraploids 4N, and so on. If you happen to see this in a textbook, this is probably the notation you'll tend to see. Most organisms that we care about are diploid. We're diploid. Most animals and plants, actually not all plants, lots of plants are not. Plants are kind of weird in that their ploidy can vary quite substantially. Most animals are diploid. But we go through this haploid stage through meiosis. So I've not got meiosis on here. You don't really need to remember the details of meiosis because this is more of a biological thing rather than something that we're specifically modeling. But in meiosis, we go from having a diploid cell that originally produces two daughter cells that have 2N chromosomes as well, and then they split themselves into two. This is important, this sort of diploid to haploid stage, because it breaks up these haplotypes here. This is a process known as segregation. Okay. We talked about heterozygosity as well. So we talked about whether an individual is a heterozygote, for example, properly. So in the case of a single loci, big A, little a, this individual is a heterozygote. So if you think about them inheriting one haplotype, or one, in this case, just one allele from each parent, this is just saying that those alleles are different. Whereas a homozygote has the same alleles. Obviously, when we get to multi-locus models, we can have individuals that are heterozygous at one locus and homozygous at another. So more generally, we can think about the heterozygosity of individuals, if you like, as being the proportion of loci that are heterozygous. The heterozygous or heterozygote homozygote distinction is important when it comes to things like fitness, especially because we might have cases where heterozygotes are more fit or less fit than homozygotes. And that can affect how quickly alleles spread in a population. It can also tell us something about the maintenance potentially of disadvantageous alleles in a population. Okay. From an evolutionary perspective, the next slide, three things are really important: fitness, selection, and mutation. So fitness you can think about as being some measure of how organisms are able to propagate their genes. Okay, so if you remember, we talked right at the beginning of the course in the first lecture about some misconceptions about evolution. We talked about, the survival of the fittest and make you think that it's all about survival. Okay, but it's not all about survival. If, say, an organism survives for 100 years but doesn't produce any offspring, well, they're not going to be able to propagate their genes. Okay, so there's some balance here between survival and reproduction that you need to be able to propagate your genes. That's something really fitness encompasses. Okay, and then selection. We focused on natural selection really rather than sexual selection. But natural selection is just this differential survival of genotypes or phenotypes in a population. Okay, so there's some heritable variation, typically when we're talking about evolution, and the natural selection is basically just acting on that heritable variation. So it's saying, okay, differential survival and reproduction between individuals leads to changes in the frequencies of alleles in the subsequent generations. And then finally, mutation. Well, we need to have some genetic variation in order for us to have selection acting on differential fitnesses. Okay, so we might have a scenario where everything has the same fitness, but there is genetic variation, in which case everything is just neutral. We could have a case where there's no mutation and there's just what we refer to as standing genetic variation. So this is essentially if the mutation rate is really, 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 really low, kind of like when we're doing adaptive dynamics, we're not actually explicitly modeling that mutation process. We're just saying, okay, there's these two types in the population, saying which one wins or what happens to them over the long period of time. We can approximate low mutation rates, and that's when we have standing genetic variation. So you can have selection when there's no mutation, but really, in the truest sense, we should be having selection and mutation together. Mutation is, you can think of as providing like the raw materials on which natural selection acts and gives you evolution. Again, remember that selection and evolution, natural selection and evolution are not the same things. Okay, this whole course we're really focusing on deterministic dynamics. And so if an allele, say, is fitter than another allele, it will spread through a population. Okay. However, if you remember on one of the homework assignments, we talked about drift, and you did some simulations of drift. We've not talked about drift in this course at all, but drift is essentially another process that can give you evolution, not by selection. Okay, so evolution is just populations changing over time. Selection is a particular process that gives you that. Okay, one of the particular types of selection that we looked at was frequency-dependent selection. And this is a case where fitness depends on frequency. So it depends on allele frequencies, one or more. And this can arise for a number of reasons, typically due to some kind of interaction. So, for example, in the case of host pathogen interactions, where pathogens maybe have to match their hosts in order to infect them, we would expect that the pathogens that are able to match the most common hosts will be able to spread, so they'll be fitter. And pathogens that match the rarer hosts are less fit, so they won't be able to spread. And there's this feedback mechanism that comes into that, which means that host alleles go up in frequency, and then they're tracked by pathogen alleles that match them, and so on. And so there's this frequency-dependent selection. That's kind of going between two populations. You can have it within a single population as well. For example, the fitness of, say, an allele that is a cooperative allele will depend on the frequency of other cooperators in the population. We met this a lot when we did evolutionary game theory, this idea of frequency-dependent selection. There's not just typically this constant fitness. Okay. We talked as well about Hardy-Weinberg equilibrium. So if we have two alleles at frequencies P and Q, then Hardy-Weinberg equilibrium essentially is just the expansion of this P plus Q squared. It's like that just equals 1 because we're talking about allele frequencies. And this tells us what are the frequencies of, in the case, suppose this big A is the frequency P is the frequency of big A, and P squared will be the frequency of AA, big A homozygotes. 2PQ will be the frequency of the heterozygotes, and Q squared will be the frequency of the other homozygotes. Okay, so Hardy-Weinberg equilibrium is, again, kind of like a null hypothesis. If there's no selection acting, then there should be no change in allele frequencies. We talked about dominance as well. So, for example, suppose our big A allele is dominant. If it's dominant, that basically means that the fitness W big A is equal to that of the heterozygote, big A little a, and this is different from that of the other allele, which is assumed to be recessive. Conversely, if big A allele is recessive, it's just the reverse of this. Okay, so in this case, we would have big A is different from big A little a. These are just reflections of each other, essentially. What's going on really with dominance is if we go back up to this genotype here, it's saying that there's an interaction between this AI and this AK. The dominant allele is the one that is expressed. These as being, we're talking about fitnesses down here, but you can really think of them as being phenotypes. So this is saying that big A has the same phenotype as big A little a when big A is dominant. Conversely, the heterozygote a big A little a has the same phenotype as little a when big A is recessive. This turns out to be really important for things like maintaining genetic disorders in a population. That's because even though something might be, many genetic disorders occur in homozygous individuals, so they're recessive. And so they're able to be maintained often because heterozygous individuals don't show any signs of disease. So they have the same phenotype, maybe same fitness as the rest of the population. And so it's not purged from the population. So this is a bit of a problem in diploids. Notice you wouldn't have this in haploids because there's no sense of dominance in a haploid population because there's only one copy of the chromosomes. Recombination turns out to be really important in a process of reproduction because it breaks up haplotypes during meiosis. Why does this matter? Well, it means that if we didn't have any recombination, say this individual up here would only ever be able to produce gametes that are AIBJ or AKBM. So they would only be able to produce two types of gametes. However, as soon as you have recombination, then that means now they would also be able to produce gametes that are AIBM and AKBJ. Okay, so recombination can break up haplotypes. That can be advantageous if it can produce novel combinations of alleles that form these haplotypes. Those novel combinations can then potentially, they might be have higher fitness, so they might be able to spread. They can also be disadvantageous. Suppose this is the best genotype, and in turn best phenotype in the population corresponding to this genotype up here. Any amount of recombination is going to produce gametes that are less fit. So potentially they're producing offspring that are less fit themselves by breaking up what might be a very favorable combination. Okay, so recombination is this double-edged sword. And then finally, we talked about linkage disequilibrium. We define this as being D is equal to X11, X22 minus X12, X21. Sorry, I don't know why Siri is appearing. So this is our linkage disequilibrium where Xij is the frequency of haplotype Ij. Anyone know how to disable Siri? Siri, please go away. When the machines take over, we'll remember this moment. I was rude to Siri. Okay. Spent quite a lot of time going through that stuff, so I'll maybe speed up just a little bit. So I wanted to go through some of the key things that we've shown. So these, in some ways, like we were really focused on very simple models of populations. We've made a lot of simplifying assumptions, okay? Real populations are not as simple as the things that we've been looking at. However, we have learned some really fundamental things about how evolution proceeds in relatively simple cases. And although in many sort of more realistic cases, the details start to matter a little bit more, a lot of the things that we've learned do carry over, and they're often a very good first approximation to what we might need to look at, okay? So, for example, we've understood that the rate at which an allele, say, spreads in a population, it's all about fitness relative to the mean of the population. Okay, we often saw this relationship of some fitness relative to a genotype, a phenotype, allele, relative to the phenotype and the average fitness in the population. Okay, so that difference often gives us some proportional rate of change. And we saw this, for example, in the Fisher-Haldane-Wright equation. We also saw things like selection coefficients and selection differentials. These tell us as well, they can be very good approximations, and we used, for example, during the pandemic to understand when different variants were coming in. You don't necessarily need a very complicated model with all the intricacies of what's going on in a pathogen and in a human population to understand selection differentials. You can use these very simple models to make a pretty good first approximations to what's happening. We also learned, I mentioned this already, Hardy-Weinberg equilibrium. I've misspelled Weinberg. In the absence of selection, diploid populations do not deviate from this. We also learned about the rate of evolutionary change being maximized when genetic variance is highest. We kind of skipped over this a little bit, but it turns out to be something that's quite profound. Essentially, if you need to have a certain amount of genetic variation for evolution to act through natural selection, and that rate of evolutionary change turns out to be highest when the genetic variance is highest. So high diversity in a population tends to lead to the highest rates of adaptation. We also looked at conditions as well for stable polymorphism. We talked a bit about rare genetic diseases. I've already mentioned this already about the fact that they can be maintained at mutation selection balance, even if they're highly disadvantageous. If they're recessive, they're very difficult to purge from a population. You can also see this incidentally, remember on one of the homework exercises that we talked about the rates at which alleles spread if they're advantageous through a population. You can flip that around and think about the rate at which they're purged from a population as well, if they're disadvantageous. And you'll see then that being recessive means that it's very difficult to purge them. We talked about how dominance affects the rate at which alleles change in frequency as well. Again, this can come from the Fisher-Haldane-Wright equation. And we talked, I just said a moment ago about linkage disequilibrium, this non-random association of alleles or haplotypes, how this can decay exponentially in the absence of any selection. This occurs due to recombination. So you remember if we had an equation that looked like this, what's our linkage disequilibrium over time? Well, it's what we start with, times by 1 minus r, where r is the recombination probability, and t is just generations. Okay, any questions about population genetics before I move on? Everyone happy? Yes.
So for linkage disequilibrium, sometimes we set the sign of D to like be positive and sometimes negative.
Mhm.
Like I didn't really like understand why, how we make that mistake.
Sure, good question. Okay, so this is how we defined linkage disequilibrium to start with. So this would say be the frequency of haplotype A1B1, A2B2, A1B2, A2B1. Okay. Firstly, I would think that it's arbitrary the way in which we've defined this. We could have defined D to be X12, X21 minus X11, X22. Okay, so that's one thing that tells us about like we've just arbitrarily defined linkage disequilibrium to be positive if this term here is greater than that term. That's completely arbitrary. We could just switch everything around. Why might it be positive? Well, if it's defined like this, that's basically just saying that these alleles these haplotypes A1B1 and A2B2, the product of those haplotypes, is just, they're essentially more common than the other versions. So A1B2, A2B1. Okay, so that's why it might be positive. And conversely, if these ones here are very common, then are much more common than the A1B1, A2B2 ones, then it will be negative. Okay, so it's all about essentially what is more common in the population. So if you suppose that these two haplotypes were much fitter in the population, then it would be positive. If these two were much fitter in the population, then you generally expect it to be negative. But it's really an arbitrary definition that we started with. Yep. Any other questions? Okay, let's move on to evolutionary game theory. So in weeks 4 and 5, we focused on evolutionary game theory. We looked at some different types of games. We talked about symmetric and asymmetric games. We talked about essentially steady states. We talked a lot about Nash equilibria and a stronger condition called evolutionary stable strategies. And we discussed a few different applications, for example, understanding competition for resources, cheating versus cooperation. We talked about balance as well between, say, producing male and female gametes. Okay, so in terms of key terminology, your payoff is essentially analogous to fitness when we're talking about evolution. Do you remember that there was originally an economic game theory, if you like, and then we just extended this to evolutionary game theory? So we're taking those ideas about rational human actions and then saying, okay, well, rather than about rational human interactions in individuals, we're translating that concept of payoff through to evolution and thinking about the payoff as being some measure of fitness within a population. And the sort of rational aspect is really what is natural selection doing? Okay, natural selection is, say, maximizing the payoff. Okay, so we talked about how we could represent this in terms of a matrix or a table. In terms of symmetric and asymmetric games, if we have symmetric games, then it's basically saying the same payoff table. So payoff matrix, I'll call it here. And strategies by implication. So examples of symmetric games might be individuals competing for resources. The resource means the same to them. They can play the same strategies. We also talked about asymmetric games. These asymmetric games could be games between species, for example. They could be between individuals that employ different strategies. So between species, for example, predator-prey interactions could be seen as an asymmetric game. Mating dynamics between males and females can sometimes as well be seen as an asymmetric game based on things like information that's aware to them, or just simple things like, you know, when they might prefer to mate. So in an asymmetric game, we would need two matrices, but we can represent this in a payoff a table as opposed to a matrix. We talked about zero-sum games. For example, the matching pennies game that we looked at was zero-sum. It's basically saying that there's a winner and a loser. So you say your payoff for player 1 is equal to minus payoff for player 2. It's slightly more specific than just a winner and a loser. They might win and lose by different amounts. Technically, a zero-sum means that their payoffs actually cancel completely. So they would sum to 1. the expected payoff that an individual would get playing a particular strategy depends on the strategy that they're playing and depends on the strategies that their opponent's playing as well. So you can think of this as being the product of this is a weighted sum based over the probability distribution of outcomes. Okay, so expected payoff depends on probability of each outcome. We talked about pure strategies, which we can represent in a two-strategy game as being a vector 1, 0 or 0, 1. A pure strategy is just saying you play a particular strategy 100% of the time. More generally, a mixed strategy would be in vector notation written as, say, P1, P2, with P1 and P2 being between 0 and 1, non-inclusive. And this mixed strategy here is kind of like everyone going around with a random number generator. And when they come to a point where they're going to be playing a particular strategy, or have to make a decision on what strategy they're playing, they play P1 with probability, sorry, they play strategy 1 with probability P1 and strategy 2 with probability P2. It's clearly just going to be 1 minus P1. And so if their random number generator is less than P1, so they play strategy 1. Is Siri coming back? Okay. So the pure strategies are essentially just a more special case of these mixed strategies. Okay, Nash equilibria. Can anyone tell me how they would characterize a Nash equilibrium? Yep.
Strategy which they would not want to change if they are having the maximum process.
Yeah, so essentially exactly, yeah, there's no incentive to switch strategy. It's kind of the key thing to remember with Nash equilibria. We did meet definitions in the case of like a symmetric game as to how we can define it based on the values in the payoff matrix, but really it's all about no incentive to switch strategy. You can also see this referred to as being it is the best response to itself. If it's a strict Nash equilibrium, then it is the unique best response to itself. If it's a weak Nash equilibrium, then it's a best response to itself. You can't increase your payoff by switching strategy, but you could switch to a different strategy and maintain the same payoff. Okay, so there's a distinction here between strict and weak Nash equilibria. We also met this evolutionarily stable strategy as well, or ESS. In the case for our symmetric games, remember when we got to asymmetric games, this kind of concept broke down and didn't really make any sense anymore. An ESS is essentially a Nash equilibrium that is uninvadable. So it's basically saying if you have a weak Nash equilibrium, there is another strategy that has the same payoff as the strategy when you play your strategy against itself. However, that one is unable to invade because it has a lower payoff. Okay, if you end up in that scenario, then it's an evolutionarily stable strategy. If you've got a strict Nash equilibrium in a symmetric game, it is automatically an ESS. Note as well that, remember we talked about these, we saw these images that looked like this. You can think of an ESS as being kind of like the top of a peak. So you can have two ESSs in, say, the same game. One might have a higher payoff than the other, but they're still locally ESSs. Okay. What kind of things did we look at? Well, we learned how to determine whether a pure strategy is the best response to itself and whether it's unique or uninvadable. So this is all about Nash equilibria and ESSs. So for example, we met this condition in symmetric games. If the payoff of strategy I against I is greater than or equal to the payoff of strategy J against I for all J, then I is a Nash equilibrium. This is nice because that means that if you've got a payoff matrix in a symmetric game, you just need to look at each column to see whether the elements on the main diagonal are greater than or equal to all the other elements on in that column. That will tell you if you've got a Nash equilibrium. We also learn how to determine if a mixed strategy is a Nash equilibrium. Remember here we have to consider expected payoffs. In the case of asymmetric games, we would have to condition on what the other individuals, the other type of individuals are doing. How likely are they to play one strategy versus another? And then give us expected payoffs. It gives us a set of linear equations that we then need to solve. Remember, when you're solving those, what we're really saying is we don't actually care at that point that we're trying to find this mixed strategy Nash equilibrium, what the expected payoff is. What we want is your opponent to basically be indifferent to switching their strategy. So that's why we just are able to eliminate their expected payoffs. We learned in a sort of more interesting evolutionary sense that optimal strategies for individuals can reduce mean population fitness. If this is some measure typically of like per capita growth rates, then it means that ultimately it's going to reduce population size. This is again, one of those like fundamental insights from a method that is very, very simplistic, but actually tells us something true about how evolution proceeds. It doesn't maximize what is good for the population, doesn't do what's good for the species. It really functions at the level of individuals, or more correctly, at individual genes. So we saw this, for example, in the hawk-dove game. So remember, this was competition for resources. Hawks fight for resources. They can potentially incur a cost because they're fighting. They might get injured. Doves just kind of display, and then one of them backs down if it's two doves. Hawk will always beat a dove. This means that there's two potential, depending on the relative costs and benefits, there's two potential Nash equilibria. There's one where the hawk and dove coexist. There's one where the hawk always wins. But regardless, if you map out the mean population fitness, it always decreases as the frequency of hawks increase. There's no scenario where doves by themselves is a Nash equilibrium. So regardless of what happens, you would always expect evolution to reduce mean population fitness. And hence, if this is a measure of growth rates, it would be population size as well. Finally, on here, we looked at also how evolutionary game theory can explain a variety of different biological problems, things like equal sex ratios. Again, this is kind of like a null scenario of, okay, if we don't see equal sex ratios, then something must be going on in terms of, say, parental investment to those to male or female offspring. This is a classic example of frequency-dependent selection. We talked about things like reproductive timing as well, and mixed strategies and antagonistic relationships. Okay, the final thing to go through today is adaptive dynamics. So we met this last week, and it builds on some of the sort of the flaws of evolutionary game theory by explicitly modeling those evolutionary dynamics rather than just focusing on steady states at the end, like Nash equilibria. We're going to be going into this in a lot more detail. So essentially, we've just had a taster of this so far. Again, there's quite a lot of terminology, but this terminology will be met again and again. So it's worth, even after the midterm, paying particular attention to this slide, maybe. So we met replicated dynamics first, and this was just a way of saying, okay, if we've got standing genetic variation in a population, we're not going to model that mutation process. We've got a number of different phenotypes of frequency pi, and they change in frequency proportional to pi, and that constant is mi minus m bar. I say constant, this mi doesn't necessarily have to be constant. This mi here and mr and m bar as well, because these are the mean per capita growth rates of the population and per capita growth rate of phenotype I. These could be functions of the frequencies of other individuals in the population. So these could depend on P1 to N. But this is a good first approximation to being able to model some evolutionary dynamics in this phenotypic sense. But when it comes to doing adaptive dynamics, we can think about just a resident and a mutant population. I'll just jump down here for now. So rather than having n different phenotypes, we can think about what happens if we're competing one resident or what gets one mutant. We do this because we're separating ecological and evolutionary timescales. So you can think of ecological timescales as being your population dynamics, if you've met population dynamics models before. Your evolutionary timescales, that's your trait or phenotype dynamics in these models. Okay, so separate these timescales means that we don't actually have to worry about mutation processes. We can just compete a resident that's typically reached a stable equilibrium against a rare mutant. That simplifies the analysis greatly, and then we just iterate this process. We made a model of interest-specific competition as well. We'll be coming back to this model afterwards. This was just a case of thinking about if Ni is the number of individuals of a particular phenotype with intrinsic growth rate Ri, then its growth rate is going to have this density-dependent term here, Aij over Nj, Aij times by Nj over Ki, where Aij is some competition, strength of competition between individuals with phenotype J and phenotype I. We sum over all phenotypes in the population, and we divide by the carrying capacity for phenotype I. This is the Lotka-Volterra competition model. Well, again, we'll be coming back to this. This isn't an equation you need to memorize. It's just to remind you, this idea of competition is really important. We can go from this kind of ecological model to an evolutionary model by thinking about separating timescales. Linear stability analysis. This is not going to be on midterm. So I'm saying that with confidence. I'm pretty sure it's not on the midterm. I have to go back and check. I will email you if it is on the midterm. I've decided to do a review based on sort of feedback from people's faces in the last lecture about not being sure about linear stability analysis. So next Monday's lecture is going to be a recap, a review of linear stability analysis. Okay, this is important because we're doing this separation of ecological and evolutionary timescales. So it's kind of a prerequisite before we're doing adaptive dynamics. So I'll go through that next week. Yeah, I'm confident it's not in the midterm. Let's see, what else have we got? Invasion fitness. So for example, our invasion fitness we refer to as W as a function of XM, XR, where X is our phenotype and the subscript refers to the mutant or the resident. This is really saying what is the fitness of a rare mutant in an environment created by this particular resident phenotype. And really all of this is just the per capita growth rate of my mutant. And really this is when NR is equal to NR star. Okay, so when it's at a steady state. We talked about the selection gradient, which is the derivative of this invasion fitness. So S of XR is equal to dW dXm divided by XM, the partial of this invasion fitness with respect to our mutant trait, and then evaluate the mutant trait at the resident trait. We talked in the last lecture about why that is. I won't go into that again now. A singular strategy, X star, is just when this selection gradient is equal to 0. So this selection gradient is telling you the direction in which your trait is going to evolve. If this selection gradient is positive, this trait XR is going to increase. If it's negative, it's going to decrease. When this selection gradient vanishes, that means it's neither decreasing or increasing or decreasing anymore. It's some kind of analog to an ecological equilibrium. Okay, so it's some steady state. We then need to think about its stability. And it turns out that this is evolutionarily stable if this quantity here given by the second derivative of your invasion fitness with respect to your mutant trait, and then evaluated with these traits at your singular strategy. If this is negative, what this is really saying is your invasion fitness here is a maximum. So this is telling you about the curvature. Just as if you're looking at some function f of x and you think about what happens to the curvature of that function when you take its derivatives, you think of its second derivatives, gives you its maximum. Yes.
Wasn't the invasion thickness partial derivative by nm of dn by dt?
Yes, but also in this particular model here, it ends up being the same thing. So usually we're thinking about its per capita growth rate. In this particular model, they turn out to be the same thing. Yeah. We'll talk about the invasion fitness more general as well in terms of what happens when you don't just have, say, one equation. We'll come back to that later on. Okay, so that gives you your evolutionary stability. One thing I will say, these equations are not on the midterm because they're derived from each other. So I would expect you to know how to get these things. Okay, so I've not included these equations on the midterm simply because they are taking derivatives of one another. Okay, so I would expect you to at least know these things. So it's not strictly a memorizing an equation per se. These are just how things are defined in adaptive dynamics. Okay, so that was one hint I would give you. Okay, we're pretty much at time, so I won't dwell on these too much. We've learned how to derive equations for our replicated dynamics from first principles. We've learned about this process of separating timescales and making assumptions about the mutations allows us to simplify the process. Again, we're going to be coming back to all these things after the midterm. And we've learned how to determine whether a rare mutant can invade and what happens in the long term. So this direction of phenotypic evolution depends on your selection gradient, like I've just said. And we did a little bit of modeling to start with on intraspecific competition. The details of this last one don't really matter, except for the process of how one finds things like a singular strategy. The actual sort of this relationship between carrying capacity and competition is not something you need to remember. Okay, conscious that this did take longer than I thought it would. Just a few tips for the exam. Please be on time. It's a 90-minute exam, but so that we don't end up with people coming and going all the time, it will start at 10:30 and finish at 12. Okay, so be on time. If you arrive late, it will still finish at 12. Okay. Do try to make your answers as clear and concise as possible, and try to structure your answers. I always talk about this idea of answers having a beginning, a middle, and an end. So you set up your problem, give your necessary definitions at the beginning, carry out your analysis in the middle, and then you state some conclusion at the end. This process generally makes it easier to award partial marks. Okay, so generally we want to try and award you marks. We don't want to just deduct them if we can't. The more structured your answers are, the easier it is to give you marks. Okay, even if you end up with the wrong, if you sort of make a mistake right at the beginning, if the process is clear that you've taken, you can end up getting a lot of the marks, even if you get the wrong answer. Okay. So this is all about showing you're working. Make sure you define everything clearly. Make sure your figures are nice and clear. No postage stamp size figures, please. And that is it. I have office hours now in case anyone has any burning questions. If not, I will see you guys on Wednesday. Best of luck.